Hello everybody. Today I will conclude the chapter on hormonal regulation of metabolism. Like I said previously, 90 to 95% of the material that you will hear today, you already know. We've discussed this in previous chapters. The only difference is that in those chapters we talked about specifically about the regulation of a specific reaction or pathway, but in this presentation you will see all of these mechanisms combined together. Hopefully that will give you more of a complete picture of what is happening in the cell. And since glucose is the main source of energy for the body and the brain specifically, I will begin this presentation with focusing on it. So the normal glucose levels in the blood is around 4.5 to 5 millimolar. Does it go above? Of course. Does it go below? It can, but not under normal circumstances. So when the glucose concentration goes above above the 5 millimolar benchmark, insulin is secreted, and the signal that insulin carries to the tissues is pick up glucose and use it, make energy, or if you don't need energy, store it, either as glycogen or as fat. If the blood glucose levels fall below this level, then another hormone is secreted, the glucagon hormone, that tells the tissues to start using the stored sources of energy, such as glycogen and fat. The purpose of glucagon is to keep the blood glucose levels normal, so that will provide necessary source of energy to the brain. So for that reason, the body will focus on breaking down glycogen and breaking down fat so that we can use it for energy, glycogen so that we can generate glucose for the brain and the rest of the tissues that need it. And also fat, in extreme circumstances, can also be used for ketone bodies production, which is an excellent replacement as an energy source for glucose. But we'll talk about that a little later. So the effect of insulin can be described, is well described in this table. You can see the glucose uptake by the muscles and adipose tissues is increased. In the liver as well. But someone might say, but the liver doesn't feed with glucose. So basically it doesn't use glucose for its as primary energy source. It uses fat. And why would liver increase its glucose uptake? Well, it increases it for multiple reasons, but one of those is, for example, to use the products of glycolysis for other synthetic purposes, such as fat, for example, or cholesterol, or even amino acids. And from there, it repackages, reforms glucose into fat, repackages and sends it out. In addition, it also picks it up so that you will store it as glycogen. The liver is the main source of glycogen, which is the major reserve for glucose in the body in case of starvation. So, increased glucose uptake. In addition, glycogen synthesis is also stimulated, as you can see in the liver. We just mentioned that. Glycogen breakdown will be decreased, of course. If you're increasing synthesis, why would you break it down? We also have an increase in glycolysis and acetyl-CoA production, again in the tissues that primarily use glycolysis. Those are muscles. Again, why would the liver do that? Well, the liver does use some of that glucose for energy, but again, it will use the products of glycolysis for synthetic purposes. And you can see that right here, increased fatty acid synthesis by the liver, and of course triglycerol synthesis in adipose tissue. So this is the effect of insulin. And more specifics, you're familiar with this, or these reactions. Insulin, how does it stimulate glycolysis? By dephosphorylating the enzyme that controls the production or the breakdown of fructose 2,6-bisphosphate. And what do we know about fructose 2,6-bisphosphate? It is a regulator of glycolysis. So if insulin dephosphorylates the protein and the protein now acts as a PFK2 that produces fructose 2,6-bisphosphate from fructose 6-phosphate, that increase in the concentration of fructose 2,6-bisphosphate will stimulate PFK1, which stimulates glycolysis, or the production of fructose 1,6-bisphosphate from fructose 6-phosphate. Now that's happened when insulin is secreted. Now, in the opposite side, when the blood glucose levels are low, then glucagon gets secreted, and what would it do? It would phosphorylate the protein, and when the protein or the enzyme is phosphorylated, the FPPase function is activated. It'll start breaking down fructose 2,6-bisphosphate to fructose 6-phosphate. The decrease in the fructose 2,6-bisphosphate concentration takes away the stimulator for PFK1, so PFK1 will decrease in function. That will inhibit glycolysis, but at the same time, the decreased concentration of fructose 2,6-bisphosphate will actually increase the function of FPPase1, thus stimulating gluconeogenesis, which is one of the functions of glucagon. In addition to that, when we have high blood glucose levels and insulin is secreted, it would also dephosphorylate the acyl-CoA carboxylase, another enzyme in another figure that we've looked before when we talked about beta oxidation. When ACC is dephosphorylated, and we also have plenty of glucose to produce acetyl-CoA, ACC will bind acetyl-CoA, converting it into malonyl-CoA. And where is malonyl-CoA used for? Or what is it used for? It is used for the synthesis of fatty acids. That's the first product in the fatty acid synthesis. You can see how very effectively the cell will convert glucose, in the liver specifically, will convert glucose into fat. At the same time, malonyl-CoA will shut down the carnitine shuttle that will inhibit beta oxidation. So let's look in general what happens in the liver under, well, when we're, like it says here, when we're well fed. In other words, when there's no, when we have well or nutritious food that is rich on glucose, amino acids, and fats. Well, as soon as we get the nutritious food, all the nutrients, glucose, amino acids, and fatty acids get absorbed in the blood and in the lymphatic system, and they get channeled primarily to the liver. Do they go to other tissues? Of course, they can go to other tissues. They can go straight to the muscle, and they can go to the other extrahepatic tissues. It's not that they don't, but I would say 90 to 95% of all nutrients that get absorbed from the intestines go into the liver. For those of you that are familiar with human anatomy, you will actually see that all of the veins, the mesenterial veins from the intestines, they all conglomerate into one vein that goes into the liver. And why is that? Because the liver will process all the nutrients that have been absorbed in the intestines. So what happens with these nutrients? Well, first, as soon as the blood glucose level rises, insulin is secreted by the pancreas. Now, the secreted insulin will stimulate all these processes that we will see happening in the liver. When glucose enters the liver, it can either be stored, especially if it's an excess and there's not much need for energy, it can be stored as glycogen. Also, it gets sent out to the brain, to the adipose tissues, and to the muscle. Someone would say, but it's already in the blood. Why does it need to be sent out? Well, it's in the venous blood that goes to the liver, and then from the liver it gets repackaged, or there's no packaging here, but re-exported in a way, now through other blood vessels, and it goes through all of the, to the arterial blood. Well, actually, it's not really through the arterial blood, but through still through the venous blood. It goes to the heart, and then it gets sent out to all the tissues now. Regardless, the liver sends it out. Let's not get complicated with anatomy here, because I may tell you something that it might not be true, since I haven't actually checked exactly my facts, so they can be precise, and I'm speaking mostly from memory right now. But whatever it is, the glucose enters the liver, it gets either stored as glycogen or sends out. It gets sent out again for the brain and other tissues. If the liver needs to degrade it, to degrade glucose either for energy, but again, very small fraction of it will be used by the liver for energy. The products of the degradation of glucose in the liver will be used primarily for synthetic purposes. As you can see, triacylglycerides that will be exported as VLDLs and then sent out to the tissues to deliver the necessary triglycerides. And if some gets used for energy, ATP will be produced. We can also generate cholesterol, etc., etc. The fats that enter, that are absorbed in the intestines, will enter the lymphatic system, some of it in the blood. It will get transported to the liver, repackaged and sent out. Some of it will get directly to the muscles, and, well, the muscles primarily. But most of it, again, goes to the liver, repackaged and sent out. The amino acids, when they enter the liver, since this is a well-fed state, if they are used for protein synthesis, great, and that's usually what they're used for. But if the body is fully supplied with energy, then amino acids, and if they are not used for protein synthesis, what happens is that they will be degraded to alpha-keto acids and then used either for, well, primarily for fatty acid synthesis and triglyceride synthesis. So it doesn't mean that you cannot convert amino acids to fat. You can. It's just the body has to be fully supplied with energy, and there has to be excess, and no protein can be used for protein synthesis. And then they have to be processed and stored, and that usually is as fat. You can see here that the pancreas plays a major role in this process. Pancreas has dual function, an exocrine and an endocrine function. The exocrine function of the pancreas is to supply digestive enzymes that are released in the intestines. They are released as zymogens. Zymogens are inactive enzymes, and these inactive enzymes get activated in the intestines, and when they get activated, they start doing their digestive role in the digestive system. The endocrine part of the pancreas is described here in these so-called Langerhans islets that have three major cell types: the alpha or A, the beta or B, and the delta or D cells. The alpha secrete glucagon, beta secrete insulin, and the delta somatostatin. And notice how well supplied with blood vessels are the Langerhans islets. And that is because these cells have the function of secreting these hormones into the blood. That's the definition of a hormone in the endocrine system. And then with that, it gets disseminated everywhere in the body. A question, though, we can ask ourselves is, how does the beta cell know when to produce insulin? Well, this is actually a fascinating mechanism. I I love talking about this mechanism because to me it's really fascinating. And that is, now when blood glucose level increases and it's above the glucose concentration inside the pancreatic beta cells, glucose enters the cell. Remember, the GLUT transporters are passive transporters. The glucose moves across those transporters based on the concentration gradient. So when glucose enters the beta cell, it gets degraded and eventually gets converted to ATP. If we have a lot of glucose in the cell, we'll generate plenty and high concentration of ATP will be reached. When the concentration of ATP reaches above certain threshold, that concentration then shuts down or closes an ATP-gated potassium channel. When the potassium channel is closed, potassium does not come out of the cell. Normally, when that happens, the result of that closure will be a depolarization of the membrane. Sorry, for a moment I kind of hesitated because I wanted to be clear on this. Normally, potassium is allowed to come out. That keeps the membrane potential positive on the outside and the charge on the membrane negative. But when that channel closes, the positive charge no longer exits the cell and starts accumulating on the inner part of the membrane, while the outside part of the membrane remains, the membrane potential stays constant, but the positive charges increase on the inside. That causes a depolarization or changes in the membrane potential. The depolarization gets passed on and it reaches a voltage-gated calcium channel. When the depolarization reach the calcium channels, they open up. And calcium, if you remember from 481, has a much higher concentration outside the cell and in the endoplasmic reticulum than in the cytosol. So as soon as that calcium channel opens, the calcium floods the cell. Someone would say, why is that? Why is that important? Well, apart from the calcium influx into the cell also produces insulin, which are basically sectioned in these insulin granules. But these granules cannot exit the cell. They cannot fuse with the membrane so that they can expel basically their content outside the cell. But when calcium increases in the cytosol, it makes it possible for the granule membranes to fuse with the cytosolic membranes and for the insulin content to be released. So that's how glucose controls insulin secretion. High amounts of glucose, high ATP, closing of the potassium channel, the positive charges cannot leave the cell. That causes depolarization of the membrane. The depolarization of the membrane triggers opening of the calcium channels that allow calcium to flood in to the cytosol. And then that increased calcium concentration makes possible for insulin granules to fuse with the cell membrane and release insulin into the blood. So there you go. Now you know how the blood level glucose control insulin secretion. Now you can think about it. What if there are problems anywhere in the signaling process? What would happen? What if we have mutated potassium channel? If the calcium channel is affected, or something else, for example, how will that affect insulin secretion? Anyway, these are just some things to think about. Now let's switch gears and think about what are the metabolic effects of glucagon. Now use this slide to think about it. Take a pause and think about it. What would be the effect of glucagon on the metabolic pathways? You already know that. You just haven't realized it. I'm going to move on though, because there's no point for me to stop. You can just pause if you want to think about it. But the effect is pretty much opposite of insulin. Increased glycogen breakdown. Why? We need glucose. It needs to supply the blood with glucose so that the cells and all tissues that need glucose will have it. As a result, glycogen synthesis is decreased. Glycolysis is decreased in the liver. Because, again, why? I mean, the liver doesn't really need energy from glucose. But why would glycolysis be shut down? Again, what is the purpose of glycolysis in the liver? To generate pyruvate, to generate acetyl-CoA primarily. And what is that used for? Biosynthesis of fats and cholesterol, perhaps even amino acids. Will that happen now when we need energy? No, the biosynthetic processes will be put on hold. So that's when glycolysis in the liver is decreased. Gluconeogenesis is increased. Fat immobilization from adipose tissues is increased. Why is that? Because there is a shift. The body is trying to use now fat deposits as a source of energy instead of glucose. And with that, of course, ketogenesis is increased as well. How do the fatty acids get mobilized from the adipocytes? You've seen this schematic before. When glucagon binds to its receptor through a cyclic AMP-dependent kinase, a hormone-sensitive lipase is activated and gets recruited through the lipid droplet. At the same time, pKa phosphorylates perilipins that help the recruitment of the lipase to the droplet. The lipase now starts degrading triglycerides to free fatty acids that get exported to the blood, bound to albumin, and then delivered to the cell to be degraded for energy in the beta oxidation pathway. Alright, so in general terms, what happens in the liver during the state of fasting? This type of liver is known as the glucogenic liver. The liver during the well-fed state is known as the fatty liver or fat-generating liver. So after a few hours without meals, the liver becomes the organ that supplies glucose for the cells that need it, primarily the brain. So when the blood glucose levels start falling below the normal, glucagon is secreted, and then that hormone controls all of these processes in the liver and in the rest of the tissues. One of the first things that's going to happen is all the deposits of glycogen will be degraded to glucose 1-phosphate that later on is going to be converted to glucose 6-phosphate. And then from there, it will be dephosphorylated to glucose and exported in the blood for the brain primarily. Something else that will keep that constant and high supply of glucose concentration of glucose 6-phosphate in the liver would be glycerol from fatty acid degradation. Triglyceride degradation, not fatty acid degradation. Triglycerides, if you remember, are degraded to free fatty acids and glycerol. Free fatty acids will go to the liver and to the rest of the body for energy. But the glycerol also will be used for generating glucose 6-phosphate and from there glucose. The fatty acids will be degraded for energy, but at the same time they will be converted into ketone bodies, which in the absence of glucose starts to substitute as energy source for the brain and the rest of the tissues. Now the amino acids are also source of energy under such conditions. So proteins in the liver or amino acids in the liver will be used, they'll be degraded to various products. Well, there are two types of products, if you remember. One type of products will go for the generation of glucose 6-phosphate and glucose for the blood, and the other types of products will be used for the generation of ketone bodies. It's not shown here, but you know that this happens because we have ketogenic and glucogenic amino acids. Whatever it is, that is the state where you start using proteins for energy, primarily indirectly by converting them into glucose or ketone bodies. So that is what happens in a fasting liver. Another schematic, which pretty much describes everything that we said about, is shown here, and I just want to go through it, even though it's pretty much the same thing, just so that you can see it a little bit better, from a slightly different angle. So during the state of fasting, like I said, the liver is the primary organ that provides energy for the body. The brain primarily, but also other tissues. Proteins are degraded to amino acids. Urea is being generated perhaps in higher concentration than normal, because under normal conditions you wouldn't degrade amino acids because they're not really needed unless you've consumed high amounts of food that has high quantities of carbohydrates and lipids and proteins. But now amino acids are degraded so that we can use them in glucose production and ketone body production. Again, it's not shown in the figure here, but we know we can generate acetyl-CoA from fatty acids. So urea, gluconeogenesis is stimulated, fatty acids are degraded to acetyl-CoA, but acetyl-CoA cannot really enter very efficiently into the TCA cycle because part of the oxaloacetate is used for gluconeogenesis. So in order to relieve the pressure on beta oxidation and to free up some CoA, acetyl-CoA is converted to ketone bodies, acetoacetate and beta hydroxybutyrate and acetone, and that now is used for energy by the brain and the rest of the body. An interesting part here that I would like to point your attention is that when we have high acetyl-CoA from beta oxidation, that inhibits the use of glucose for energy by inhibiting pyruvate dehydrogenase and stimulating pyruvate carboxylase. And you remember that, and we've actually talked about that when we talked about glycolysis regulation and how a higher concentration of acetyl-CoA stimulates actually gluconeogenesis and inhibits glycolysis so that it will preserve glucose and stimulate glucose production. This is a very interesting figure that shows the relative change in concentration between fatty acids, glucose, and ketone bodies. You can see that during starvation, fatty acids in the blood doesn't really change. So if you're planning to fast so that you will decrease the fatty acid concentration in your blood, that's not going to happen really, because it's not, fasting doesn't really control the concentration of fats in the blood significantly. However, you can see that as soon as you start fasting, the blood glucose level starts decreasing, and the ketone bodies are in a close relationship in a balanced relationship between glucose. So when the glucose concentration starts falling under a certain threshold, we can see that the ketone bodies production going from zero basically at first starts increasing slowly. You can see it how it's a little bit, the slope is a little bit smaller, but then after about a day, and especially after the second day, the production increases dramatically, and then it plateaus at around 6. Some might say, why does it plateau? Well, remember, if you have too much of ketone bodies, then that leads to ketoacidosis. And there, I guess a balance is reached between, in a healthy body, between the use and production of ketone bodies. But I actually just today realized something about this figure. There was a time when I watched this documentary about fasting, and as a way of treatment of metabolic conditions, and diabetes or cardiovascular disease or other conditions. And there was this documentary I actually watched on Amazon Prime that talks about the science of fasting. Actually, I believe that's the name of the movie. If you want to watch it, it's very interesting. The science of fasting. And all people say that these are people that will do extensive fasting for days, weeks, as a matter of fact, just water. And all of them said that the hardest part is the first 3 days. So right up to here. And after that, I mean, you're tired, but it's not difficult anymore. It's very difficult to fight hunger and that drive to eat up to about 3 days. And I remember that. But it was interesting what I saw here, that this table, this graph shows that at approximately the third day, there is a switch where basically the ketone bodies now have much higher concentration than glucose. And while I'm not saying that the body does not depend on glucose anymore, perhaps the body switches. And now the drive for hunger, for example, or the primary source of energy being now the ketone bodies, hence fats and the fat deposits from the body, no longer requires to stimulate the appetite to stimulate appetite. And for that reason, perhaps people don't feel hungry after the third day. Now, I want to say that this was just speculation on my end. I don't want anybody to start saying that that really is the case. For me, I just wanted to share something that I realized today. Whether it's true or not, I don't know. But it's an interesting overlap. Like I said, fasting people usually say that after the third day, it's no longer hard for them to fast. Perhaps that has to do with the ketone body concentration in their blood, and perhaps the body switching and saying, now from now on, I will depend primarily on ketone bodies, and I will not care so much about glucose. And as you know, the only source, the main source of glucose in the body is food. Our deposits of glucose only last for about 12 hours, the glycogen deposits. And after that, we have to start depending on fat and proteins, which generate glucose, but also generate ketone bodies. So perhaps there is a switch that happens. Now the body starts depending primarily on ketone bodies. Anyway, it's just an interesting observation. All right. Now at the end, I also want to mention a couple other hormones in addition to insulin and glucagon, because they have a major metabolic effect, but they are not directly related to metabolism. Their role is not primarily metabolic regulation. One of those is epinephrine or adrenaline. This is a hormone, as you know, that controls fight and flight. Basically, it tells the body, get ready for action, like it states the figure, the table here. So physiologically, increased heart rate, blood pressure, dilation of respiratory passages, all of that, the goal of all of that is to increase oxygen delivery to the tissues, primarily the muscles. Think about it. If you're anticipating something, let's say you're at the start line of a race, what happens? Your heart rate starts pumping, you're breathing heavily, your blood pressure rises, and that is all in anticipation of an activity. So metabolically, what would the body want? It would want energy, fast energy. What does that mean? High blood glucose concentration. So glycogen breakdown will be stimulated. Glycogen synthesis will be decreased. Gluconeogenesis will be stimulated. Why? The goal is to increase glucose in the blood because that's a fast access of fuel. At the same time, the glycolysis in the muscles is increased. Again, why? ATP production that is used where? In muscle constriction. Glucagon secretion will be decreased. Why? Glucagon secretion will be increased because what is the purpose of glucagon is to increase blood glucose. So that's why glucagon is increased. Insulin is decreased. And so that's why I sometimes draw this correlation that, or some people have drawn this correlation, this is a matter of fact, that it is not good for diabetics to be stressed out, to be agitated. Why? Because adrenaline raises the blood glucose levels. So that's why if you're diabetic, you should try to stay calm and not get stressed out too much, because adrenaline is a stressor hormone. It gets released under stressful situations, and it would raise your blood glucose levels, which will make diabetes worse. Just a few thoughts about this actually very interesting hormone. And at the end, I also just want to briefly mention the role of cortisol, and mainly to make one point that this is a long-term stress hormone that has a long-term effect. His primary role is to restore blood glucose level concentration and to increase glycogen levels. I'm asking you here, do you expect the response of cortisol signaling to be fast or slow? Well, I mean, you already hear that this is a long-term stress response. But there is something else also that you need to realize to really answer this question correctly. Do you expect for this cortisol or corticosteroid hormone to have fast or slow response? You have to make the mental connection that this is a steroid hormone. And steroid hormones, they transition through the cell membrane and bind receptors in the nuclear membrane. And what do we know about signaling through receptors that are at the nuclear membrane? That those signals are slow. And why are they slow? Because they control gene regulation or gene expression. And that's really how cortisol works. It controls amounts of enzymes. It controls expression of enzymes. And for that reason, the response is slow, but it's long-lasting, because it doesn't just affect temporarily how an enzyme would function. It affects how much enzymes or how many molecules will be expressed, how many polypeptides or proteins will be expressed in the body, and how do they function. And it would also change the expression patterns. And for that reason, long-term high cortisol concentration can have dramatic changes, can result in dramatic changes in the body. Primarily affects the muscles, it affects the fat deposits, the calcium deposits in the bones, endocrine function. This hormone is a very treacherous hormone, widely used as a therapeutic because it suppresses the immune function. So it's used as immunosuppressant, but at the same time, it has severe and drastic and long-term effects on the body in general. All right, with that, I will conclude my presentation. I hope this was interesting. Like I said, pretty much nothing that you've seen in this chapter is new for you. I'm sure it's going to be interesting. But If you have any questions, please let me know. And you can come and see me during my office hours, and I'll be happy to talk to you.